A very good afternoon to everybody. I have no financial disclosures. Every year, 35 lakh babies are born preterm in our country. Even if we assume the utopian situation where all these babies are screened, it will mean more than 17 million eye examinations. For this rural baby from Gulbarga, it would mean traveling 5 times, 250 kilometers each, only to be told that she does not have ROP. The problem is ROP is multifactorial and we don't really know how to predict which baby will get the disease. Some of our low-cost models that we are using in rural Karnataka are not good enough because we still can't predict which baby will have the disease in the first visit. Hence the elusive search of a so-called biomarker. Why not tears, we asked. After all, tears are being used as biomarkers in systemic conditions and even in some adult ocular disease. In the past few years, the dramatic rise of publications in this area is amazing, but yet there are none in the field of biomarkers in infants in tears. Therefore, this study, where we aim to check, to first of all detect, if it's possible even, from tears in the first visit, and see if we can find a biomarker. About 39 preterm babies with and without ROP were included, and while they were undergoing ROP screening, they underwent tear examination, tear assess, through using the Schirmer's method in several visits. We made sure that the height of the Schirmer strip was 20 millimeters or more. These strips were then taken using the cold chain into the cryo-freeze of our laboratory, where they were archived and processed. Strips were cut into small bits and then put into an agitator with PBS solution at 4 degrees for about 2 hours. The analyte was then taken and put into a centrifuge, and this is where we placed those tiny antibodies for all the antigens that we wanted to detect, which I'll come to in a minute. And finally, the flow cytometer not only detected these antigens, but also quantified them so that we got a report. Now, these are the 10 antigens or proteins that we looked for. Now, these are Greek and Latin to all of us. It's not important. What's important are what the families they belong to. The first one are those that cause angiogenesis, VEGF and angiogenin. The other one actually attracts cells to cause angiogenesis, the chemoattractants. Then cytokines actually talk to each other, information between cells. And finally, those that adhere to the endothelium budding as they progress forward. So these are the 10 antigens that we looked for and the reasons why we chose them. Now let us look at these results as a clinician would rather than a scientist. These babies went from no ROP to no ROP, which means they were the controls. Their blood vessels grew from immature to mature, reaching the edge. Now what's interesting is in the very first visit, when they didn't have anything, they had in their tears higher levels of VEGF and angiogenin and all the cytokines. Let's compare it to a group that had no ROP in the very first visit, but after a few visits developed some mild disease. Now mild disease we call type 2, we don't need to treat them, but it is ROP. And the interesting thing is in the very first visit we found lower levels of VEGF, angiogenin and the cytokines. Let's move now the focus to those babies who went into severe ROP that required treatment. Now when they moved from mild to severe ROP, their VEGF angiogenin, one lesser known of the evils, actually rose, and we were probably able to predict ahead of time. When you're talking about babies who regressed and babies who progressed just from one stage to the other, again, they were diametrically opposite from their tears that we could predict. This schematic shows us what happens in the last visit. That line out there is the ora serrata. The blood vessels have reached there. This is a mature baby. But in the penultimate visit and final visit, we found a twofold rise in these VEGF factors, which means that that push is required to become mature and normal. I think the study brings out an extremely important point, a dogma that we've been holding for decades, that a preterm animal is born with low serum VEGF, and I think this is the first human molecular evidence. Interestingly, it's coming from tears. We also know now that we can assay these tears easily, detect these antigens, and actually predict which baby will have the disease and not in the first visit, and look at the interplay between the inflammatory proteins. Are we finally at that point where we can find a biomarker? Our institute is now developing a point-of-care kit. It looks something like this. We put in the Schirmer strip, and we get a readout of all the antigens that we want. What we want to do is check out the impact in the community. We have a large-scale ROP program that runs in the community, and we're going to have these point-of-care kits in there, where the mothers who are coming in with their babies are going to be subjected to tear analysis in the first visit. so that we can better prognosticate their follow-up and even predict which baby can possibly require tighter follow-up, which might need treatment. For this index baby that I showed you in the beginning, the Gulbarga baby, it would mean probably saving 3 visits. But for our country, with 35 lakh babies who need 17 million exams, we'll probably predict and prevent ROP in more than 10 million of these babies across the country. And that is what we want to do: translate research from the science, from the laboratory, into the clinic, and eventually into the community. Thank you.
What is the percentage of your false positive results in these cases?
Yeah. Automatic bead array analysis, we are now trying to only detect. The only controls that we've had are preterm babies. As we know, we don't really have a normative data to talk about which is false positive. Biological false positives are possible. So in fact, 39 babies, when we submitted the abstract, that N value has gone up to about 175. And the false positive, from what we know from the true positives, is about 8 to 9%. But I think that's going to come down as we get more normative data.
Do you have any problems in collecting the samples? The sample is that any problem like inadequate sample quantity, or the second question is, how do you find the thing is cost effective when you are talking of mass screening?
Sure. First question first, sir. Difficulties in collecting the tear sample. Believe me, we all thought that preterm babies don't produce tears. We thought the lacrimal gland starts functioning later. When we spoke to the scientists and looked at anatomical literature, it may not actually be from the lacrimal glands. It's basal secretion, we know, but a lot of it is what they call transcytosis. So some of those cells are actually migrating from the posterior segment into the anterior surface, into the cornea, into the epithelium. Now, these are, of course, not borne out from human studies, but from animal studies. So that's why I showed that video where we could actually pick up heights of 20 millimeters or more. I mean, really not a dry eye case. And as far as cost effective is concerned, yes. So I think right now the cost of the entire antigens that we want are of course shared, because in our institute several projects go on, but it's about 7,500 rupees to buy the antigens to do that. Yes, to make it cost effective, we need the point of care kit, which is what I wanted to end with, because in our institute we're doing studies on keratoconus and dry eye and glaucoma. So it's going to be one kit, and all the antigens that we want are fed in. So when you share the cost will come down, and we hope that this will eventually become something that we can use out there in the community.
At what age, that means at duration or intervals, the sample is collected and processed?
Yes, ma'am. So we are following the national guidelines for ROP. So they say the first screening should be done between the 3rd and 4th week. That's been modified by Government of India. First screening before discharge. So the youngest baby whom we have assayed for tears is 6 days old. And we do that, we are taking the tears every 2 weeks and sometimes every 1 week, depending on the diagnosis. So if it is some ROP, it would be 1 week. If it is no ROP, it would be 2 weeks, exactly the way the screening protocol dictates according to Government of India.